First of all, I would like to thank Peter and the Society for this wonderful invitation to talk to you today. It's a very big honor to speak to you, and particularly in these very beautiful settings. So, I just start with telling you very little bit about myself. I was born in Malta. It's a tiny Mediterranean island just south of Sicily. I grew up over there. I went to university over there. I first studied pharmacy, did my undergraduate project in genetics, got very fascinated by that, and ended up doing a PhD degree in genetics, which I also obtained from the University of Malta. However, this is the island, as seen from the plane, and it's not far, it's close. Which means it's very small. So the whole island is 316 square kilometers. Very, very tiny place. So, of course, opportunities are limited. And as I grew up, I was also looking outside. Where can I go? What can I do? And that is what brought me to Holland. So I arrived in the Netherlands almost exactly 20 years ago. Next January it will be 20 years that I'm here. And I was very lucky to join, at that time, the Department of Experimental Cardiology at the AMC, where Professor Arthur Wilde was at that time starting to build a new team within the department, which is molecular cardiology, with the aim of understanding the molecular and genetic basis of the normal cardiac rhythm and susceptibility to sudden cardiac arrest. So this is the topic that we've been studying, sudden cardiac death. Why is this topic important? It's important because sudden cardiac death is a very important mode of mortality in the general population. So 1 out of 5 of the individuals in the general population will die suddenly. In the Netherlands, this translates to 300 deaths per week by sudden cardiac death. And this is the arrhythmia that causes sudden cardiac death. So what we have here is the ECG, which is normal. Then comes an extra beat, which we commonly refer to as a skipped beat, but is actually an extra beat. The heart rhythm corrects itself, returns to normal for a while over here. It deteriorates again into a non-sustained arrhythmia, which corrects itself again, but ultimately it deteriorates into ventricular fibrillation. This arrhythmia is lethal unless it is removed by virtue of an electric shock within minutes. And that is the arrhythmia underlying sudden cardiac death. So what we see here is a normal beating of the heart. You see a very ordered conduction of the cardiac electrical impulse throughout the heart, which leads to a very ordered contraction of the heart. Here we see the heart beating very efficiently, pumping blood throughout the body. And this is the normal ECG. Now this is what happens during ventricular fibrillation. Now the electrical activity is very chaotic, and because the electrical activity is chaotic, the heart is not beating synchronously, but rather it quivers. The heart is quivering, and almost by doing so, it is standing still. It is not pumping blood throughout the body. And that is evidenced by the ventricular fibrillation on the surface of the body, on the ECG, on the surface of the body. So, as I said, ventricular fibrillation, one can survive an episode of ventricular fibrillation only if an electric shock, a defibrillatory shock, is administered within a few minutes. This means that only a few people survive an episode of ventricular fibrillation, either because many times also it happens when individuals are alone, or else help does not reach them on time. Now, because survival is so poor, prevention is key to dealing with this important problem of public health. And what is important is that prevention is possible, either by means of drugs, drug therapy, or or by means of an implantable cardioverter defibrillator that individuals who are at risk of ventricular fibrillation carry inside their body, which will actuate an electric shock when it is necessary. So, if prevention is key and prevention is possible, it means that it becomes very important to be able to determine who is at risk. We need to be able to know who is at risk in order to implement preventive measures on time. So here I'm going to show you a small film, and I'll show it twice because it goes very quick, of when a preventive measure has really saved a life. Sorry about that, I need to go one back. So this is a young player, 21 years old, playing in a Belgian league soccer game. You need to keep your eye on this individual on the back over there. All of a sudden, he collapses. Yeah. And after a few moments, he regains consciousness. So what happened to this individual is this: he collapses, his ICD prepares to shock. You will see the shock now. The ICD has shocked, his heart starts beating again, and he regains consciousness. So this individual was lucky enough to have been recognized to be at risk and was treated pre-symptomatically. So what has happened over there? The ICD has detected the abnormal cardiac rhythm. It charged while in the body. It prepared to shock, and it discharges shock, and the person, the heart started beating normally again, and the person regained consciousness. So now, how does a heartbeat arises? How does a heart beat? So every heartbeat is born in the sinus node region, which is a very tiny area of muscle in the heart, in the upper right chamber of the heart, which is called the atria. So there you get the birth of an electrical signal. That electrical signal subsequently travels from cell to cell through the wall of the atria to reach another node, and that is the atrioventricular node. When the electrical signal reaches the atrioventricular node, it slows down a bit, and it does this for an important reason, that it needs to give time for the atria to pump blood into the ventricles. Once the electrical signal exits the atrioventricular node, it travels very, very fast through the special wiring of the ventricles and the peripheral conduction system to activate the ventricles, which then contract synchronously to pump blood throughout the body. And this electrical signal can be evidenced on the surface of the body by means of the ECG. The ECG is designated into different intervals and peaks, like the P wave, the QRS complex, the T wave, and these intervals and the morphology of the ECG tell us about the electrical conduction in the heart. So for instance, the PR interval represents the time it takes for the electrical signal to travel from the atria to the ventricles. Whereas the QRS complex represents the time it takes for the electrical signal to travel throughout the ventricles. Whereas the QT interval is the time it takes for the ventricles to recover. So this is the ECG that I've just explained. Now, what is at the basis of the ECG? Where does the electrical activity in the heart come from? The electrical activity in the heart comes from the complement of ion channels that each and every cardiomyocyte, heart muscle cell, in the heart contains. So in the wall of the cardiomyocyte there are ion channels. These are proteins that form channels through the cell membrane through which ion channels can pass. And these ion channels are important for charge going into and out of the myocyte, which depolarizes the myocyte, taking it from a normal resting membrane potential, which is negative, to a positive membrane potential. So what we have is phasic change in the potential of the cardiomyocyte from negative to positive with every cardiac cycle. An important thing happens during this phase of the action potential. What you see here is that the action potential is maintained at a positive charge for a few tens of milliseconds. That's important because at that point, calcium enters the cell, which triggers contraction. And this phenomenon is known as excitation-contraction coupling. So this is the interplay between the electrical activity of the heart and the beating of the heart. So now we go back to the topic of sudden cardiac death. Now, sudden cardiac death unfortunately strikes across all ages in the JAM population. Most of the time it strikes at older age, where 1 out of every 1,000 per year are at risk, but unfortunately it also strikes the young, although over there the incidence is lower. In the young, it happens in the setting of inherited cardiac diseases, which have classically been divided into two types. The primary electrical diseases, where the structure of the heart is normal, and the cardiomyopathies, where the structure of the heart is abnormal, as you can see over here. So over here, the abnormal structure of the heart is at the basis, forms an important role in triggering potentially lethal arrhythmias. On the contrary, in the primary electrical diseases, the problem lies in the electrical functioning of the heart. These are the diseases which are associated with sudden cardiac death in individuals typically less than 40 years of age. In the older segment of the JAM population, on the other hand, sudden cardiac death happens most typically in the setting of coronary artery disease, when the arteries of the heart narrow and leads to ischemia, loss of oxygen in certain areas of the heart. Now one can imagine that because this coronary artery disease occurs later in life, whereas these disorders are already present from birth, while both genetic and environmental factors play a role in both, one could imagine that we think that genetic factors have a stronger contribution to risk of sudden cardiac death in the young as opposed to the old, where here you need to accumulate a lot of environmental insults to display the disease. So although our attention in the last years has also gone to for the identification of genetic factors that contribute to risk in the older segment of the JAM population, most of our work so far has concentrated on sudden cardiac death in the young. And we believe that besides helping the segment of the population, these disorders could also teach us about fundamental mechanisms of cardiac arrhythmia that also could inform us about mechanisms in the older segment of the JAM population where the genetic underpinnings are more difficult to track. So the primary electrical diseases, as I said, display a structurally normal heart, and the basis of the disorder lies in the abnormal electrical function of the heart. So it's not surprising that actually they can be diagnosed by specific features in the electrocardiogram. So what we see here is sick sinus syndrome. This is when the sinus node is not functioning normally but fires irregularly or slow. And what you see here is that the cardiac cycle is longer. The RR interval is prolonged. Over here we have an ECG of conduction disease. Here you could have conduction slowing in different regions of the heart. Either the signal travels slowly from the atria to the ventricles, where you get prolongation of the PR interval, or slowly within the ventricles, where you get prolongation of the QRS complex. Another disorder is the lung QT syndrome. You'll hear more about that later. And this is when the ventricles take longer to recover, and that is associated with a prolongation of the QT interval on the ECG. On the other hand, the cardiomyopathies are associated with a structural defect in the heart, and these can be diagnosed by means of an echocardiogram. So what we see here is an echocardiogram of a normal heart. This is the wall, the septum between the ventricles, the two lower chambers of the heart. And what we see over here in hypertrophic cardiomyopathy is that the septum is thickened. The heart muscle over there is thickened. And this is very pro-arrhythmic. It is an important substrate for arrhythmia. In the last 15 to 20 years, there has been a huge increase in knowledge of the genetic underpinnings of these two disease categories, the primary electrical disorders and the cardiomyopathies. And essentially, the fact that these disorders cluster in families has helped us a lot with tracking them down. So this is a hypothetical family where the grandmother was affected, her two daughters were affected, and subsequently their grandchildren also displayed signs of the disease. And what you do over here, you track that part of the genome, that part of the chromosome that is segregating together with the disease in the family. And by means of these genetic studies in families, we were able to identify mutation in quite a number of genes, and thereby being able to help these families. But now I'll take a little bit of a sidestep and tell you a little bit about the human genome. So the human genome is composed of 3 billion base pairs, as most of you know, organized in 23 chromosomes, 22 autosomes, and the X and Y sex chromosomes. These 3 billion base pairs are compartmentalized, so to speak, into that small part of the genome that actually codes for proteins. It holds the code for proteins. And these are the exons. And collectively, the coding region of all the genes, which only represents 1% of the genome, is called the exome. The rest of the genome, the remaining 98 to 99%, is actually non-coding. For a long time, people were calling it junk, and now we know more and more that that's not junk at all, but actually holds a lot of regulatory variation that controls the fine-tuning, the level of expression of the 20,000 or so genes in our body that code for the proteins. Two important developments have happened that have increased drastically our knowledge about genetic variation in the human population. One of them has been the drastic reduction in the price of sequencing the genome. So what we see over here is the price of sequencing the genome the first time the genome was sequenced. So the first time the genome was sequenced, it occurred in the setting of the Human Genome Project, which was a huge project taking place in 20 different labs across 3 continents and cost $2.7 billion US. The first draft of the human genome was ready in 2001. Nowadays, in 2016, you can sequence a human genome with approximately $1,000 US in one's own lab in a couple of days. And this revolution is caused by the technological advancement that happened around 2008, which is called next-generation sequencing. And many times you see the slide, which is actually making the comparison with Moore's law. What you have to note over here is this is actually a log scale. So what you see in 2007, 2008, the pace of reduction of the price of human genome sequencing far outpaced what is predicted by Moore's law. So that's one important development. Another important development, which happened secondary to the falling price of human genome sequencing, is large-scale projects of sequencing of human populations. For instance, the 1000 Genome Project, the 10K UK project, now Genomics England. In Holland, we have the Lifelines project. All these projects are sequencing vast numbers of individuals, healthy populations, specific disease groups, which are telling us more and more about genetic variation in the general population. So what do we know? There's a lot of variation in the general population. So when this was last accessed in April 2016, there was 154 million sites that demonstrate variation between us. And if we compare the genome of each of us with a reference genome, we would see that we each have 4 to 5 million variations as compared to the reference genome. Some variation arise de novo, they arrive for the first time in us, when the gametes are being formed from our parents, for instance, but the rest have been transmitted to us by our parents. So, this knowledge about genetic variation has also told us that genetic variation displays differences in frequency. Some genetic variation is very common in the general population, whereas other is very rare, and some genetic variation has a very strong effect size. That is, if you have it, you have a big chance of having the disease or a particular feature, whereas other genetic variation has a low effect size. Now, going back to those mutations that cause those diseases, causing sudden cardiac death, so diseases present from birth, we find that these are present in the segment of this graph over here. So these mutations causing these diseases are very rare in the general population and have a very strong effect size. That is, you have them, you're very likely to display signs of the disease. So where are these mutations? These mutations are in the primary electrical diseases. The structure of the heart is normal. The defect is in the electrical component of the heart, so it's not surprising that these mutations actually affect the ion channels in the cardiomyocyte, which are the conductors of electrical signaling in the cell. On the other hand, in the cardiomyopathy, there are two different classes of proteins that are affected, both structural. In hypertrophic cardiomyopathy, for instance, it is the proteins that form the contractile machinery in the cell that are affected, whereas in another form of cardiomyopathy, called arrhythmogenic cardiomyopathy, it is the proteins that mediate the contact of cardiac muscle cell to cardiac muscle cell. So this knowledge that has been gained in the last 15-20 years actually resulted in a new discipline in medicine, which is called cardiogenetics. And what I show you over here is the growth in patients seen per year at the cardiogenetics clinic of the AMC. So this is when it started, this is when these genes were being discovered, and we've seen a growth in patient numbers that has now plateaued. There are also some particular mutations that are quite prevalent in Holland. And these are called founder mutations. So what we see here is a specific founder mutation in phospholamban, which is a protein that is very important in regulation of calcium in the cardiomyocyte. What we realize is that this mutation is quite common in the Netherlands, and actually it clusters in the north of the Netherlands. And we think that it resulted a few centuries ago by a founder. So a founder would be an individual that either moved to the north of the Netherlands from somewhere else, and then his progeny carried the mutation to the present day, or else a founder might have been the one where the mutation occurred de novo in him, as I have just explained. In an effort to map this more specifically to the north of the Netherlands, so what you see here, these are the present day carriers of this mutation. So for instance, in this region, in the Pars regions, there are the most carriers identified this day. We also mapped where their grandparents are come from, because that takes us one generation up. And that then shows us a focus over here. So we think that this is probably the location where this mutation originated in the 16th or 17th century. So what I told you is that you can actually diagnose these diseases either by the ECG or by an echocardiogram. So why do we need to do genetic testing? We do genetic testing for a number of reasons. First of all, sometimes the disease is not very clear. It's not very clear whether the patient actually has a disease or not. And actually this genetic test can tell you with more certainty that the patient is actually at risk. So what we see over here is a distribution of the ECG QT interval. So this is the QT interval over here. It's the time it takes for the ventricles to recover from their cardiac electrical impulse. And what you see here is a distribution of the QT interval in the JAB population. The solid line is males, the dotted line is females. Females tend to have a slightly longer QT interval than males. And what you see over here is a distribution of the QT interval in patients with a long QT syndrome who are known to have a mutation in a cardiac ion channel. And what you see over here is that there is actually an overlap. There is an overlap in QT interval between the JAB population and the controls. And we also know that these individuals, in spite of having, at this point that this ECG was done, a normal QT interval, they're still at risk of sudden cardiac death. So in situations like this, clearly a DNA test can improve diagnosis of these patients. Another important reason to do DNA diagnostics is because we have realized that mutation in gene A may be associated with different triggers for arrhythmia as opposed to mutations in gene B. So what we see here are 3 different genes that have been linked to the long QT syndrome. So between them, these account for 90% of patients. And what we know now, because of genotype-phenotype correlation, so by relating the genetic defect with the clinical presentation of the patient, we know that swimming and exercise is an important trigger for arrhythmia in the case when the KCNQ1 gene is mutated. On the other hand, long QT syndrome patients with mutations in this gene, the KCNH2 gene, are mostly at risk, for instance, when the alarm clock rings, auditory triggers, or else females are also at risk during peripartum period. On the other hand, mutations in this gene typically lead to arrhythmia at rest or during sleep. This is not only important for lifestyle adjustment, so avoid swimming, avoid exercise, remove the alarm clock, you have to sleep. But it's also important because it can also give us a hint for gene-specific therapy. So for instance, therapy that reduces the heart rate is probably more beneficial here as opposed to here. Our heart rate goes down during sleep, so we shouldn't suppress it further because that could be pro-arrhythmic. Another rare scenario where genetic testing is vital is in the case of a small minority of primary electrical diseases that actually are associated with a normal ECG. This we call idiopathic ventricular fibrillation, and in the Netherlands, quite a bit of it is caused by mutation in this gene. Again, here we see a distribution of mutation carriers of this gene in the Netherlands, and we see that this is clustering now in the middle of the country around Utrecht. But for this disease, really only a genetic test is possible to predict risk. Not only can we use, of course, genetic tests for predicting risk, but also finding these genes has importantly given us a handle to understand the mechanisms of these diseases. So if we know the gene, we can design experiments to help us understand what happens to the electrophysiology of the cell, what goes wrong, what triggers the arrhythmia. And the workhorse for cardiac electrophysiological studies has been the patch clamp technique. Now, I feel a little bit uncomfortable describing patch clamp and electrical machinery to physicists, but nevertheless, I will give it a try. So, as I said, patch clamp is the workhorse of cardiac electrophysiology, and what we do with the patch clamp technique, essentially, we have a cell over here, it is expressing either a normal ion channel, or we put in the ion channel of copying the patient which is mutated, and then we expose the cell to a potential across the membrane, and then we measure the current that is going through the channel. And by comparing the current going to the wild-type channel and the current going through the mutated channel, we hope to understand more about the disease. So this is an example of a mechanism of long QT syndrome in this case. So what we have here is a trace of sodium current entering a cardiomyocyte, for instance. What you see over here, the sodium current enters very fast in the cell, and then by virtue of the fact that the channel closes very rapidly, the current goes back to zero very fast. Now, what happens when you have a mutation in the ion channel in a patient with long QT syndrome? Current enters the cell very fast, no problem over there, but what happens, the inactivation process does not happen normally. So to speak, the channel remains leaky, and it conducts an inward sodium current during a phase of the action potential when it should actually be closed. Now what happens is, too much positively charged sodium ions are entering the cardiomyocyte, which means that it will take longer for the cardiomyocyte to repolarize and for the action potential to return back to its negative resting state. And this prolonged action potential is visualized on the surface of the ECG by means of prolonged QT interval. So this is just to give you an impression of how we can understand how a gene mutation actually gives you abnormal cardiac electrical function. And as I said, it provides us a handle to understand the mechanism of these disorders, which is necessary to be able to identify new therapies. So for instance, understanding this mechanism allowed researchers to set up screens for drugs that actually block this late or sustained sodium current. And the first one that was identified was ranolazine, the second generation one was this compound, and now this one over here is actually in a phase 2 clinical trial at centers worldwide. So I've told you a lot about success stories, how many genes have been identified, but there are still considerable challenges. Now, one important challenge is that even if you identify the genetic defect in the family, like this one over here, not everybody who carries the genetic defect in the family is affected to a similar extent. So this plus over here represents the mutation, which is causing the disease. So in this case, inherited from the affected grandfather. What we see over here, his daughter has inherited the mutation and has the disease. But his other daughter has inherited the mutation but does not have the disease. Whereas her son has the mutation and has the disease. So what we see is a very large variability in disease severity in spite of everybody having the familial genetic defect. Now, one way of best way of appreciating this variability is by looking at large families carrying founder mutations. I show you here another founder mutation. This one again originating from the north of the Netherlands is the 17 insertion D mutation in the sodium channel. So this is a pedigree that we have linked back to the 16th, 17th century by means of genealogical research that we've conducted in the north of the Netherlands. And we've identified so far 166 carriers of this mutation that are all linked into this pedigree. And everybody with a black dot over here is carrying the mutation. And what I show you over here is the variability in disease among these 166 people. So one feature of the mutation is the QT interval. So mutation carriers have a prolonged QT interval, as we can see over here. So these are the non-carriers in the family. These don't have the mutation. Their QT interval is generally all below the normal, which is here 440 milliseconds. And as expected, most of the mutation carriers are above, have a QT interval above normal. But not all. Actually, some of them have a QT interval within the normal range. And of those that have an abnormally prolonged QT interval, some are more severely affected than others. Furthermore, if we now focus on the mutation carriers, not everybody gets the arrhythmia. Some of them remain without symptoms throughout life, whereas other ones can get symptoms already at young age. So actually, the way we're thinking now about these disorders is different from the classical view. So classically, we've been considering these disorders as monogenic. You inherit a familial mutation, you will get the disease. The more contemporary view is actually that these diseases are probably near monogenic in that you have the primary genetic defect, which determines a large degree of the risk, but you need to inherit additional genetic factors that ultimately determine your individual susceptibility to risk. And going back to the different types of genetic variations in the giant population, while the primary genetic defect is sitting over here, these modulatory genetic variants are likely to be low frequency, so less rare, more common variants that have either an intermediate effect or have only a small modulatory effect. So one outstanding problem is that variability, but another outstanding problem in the field is that actually some of these disorders have defied genetic elucidation in spite of years of research. One of these disorders is the Brugada syndrome, and you can see that over here. This is a typical ECG of Brugada syndrome, which is ST segment elevation in the right precordial lead. So it is essentially this signature over here. Brugada syndrome typically leads to arrhythmia in males around 40 years of age, and the arrhythmia typically hits at night. The only gene actually that's reliably associated with the disorder is again SCN5A gene, which codes for the sodium channel in the heart. However, in the great majority of patients, the underlying genetic defect is unknown. So classically, we've been thinking of Brugada syndrome as being familial because we've noticed in some families familial clustering. But as more and more patients have been seen, we've realized that actually many cases are sporadic, that is non-familial. So you would have one person from a family, but no other family members who are affected. Also, when we do have families, and when we do find this gene mutated, we notice very low penetrance, which means that many of the carriers of the mutation do not demonstrate the disease. And also when they demonstrate the disease, there is a large variation in expression. So this changed the way we think about Brugada syndrome and also other disorders that have defied genetic elucidation, and that they likely are not monogenic, but most probably are oligogenic, where you would need to accumulate a number of genetic hits to reach the threshold to demonstrate signs of the disease. As explained for these variants over here, genetic variants that contribute to this oligogenic or polygenic paradigm of inheritance are again either common variants in the general population that have a small effect, or as low frequency variants that have a moderate effect on risk. So in the last few years, we've started testing this paradigm for these disorders, and because it has been cheaper to do so, we started by looking at the common variants. In order to approach this research goal, you need a large number of patients, and now the study design is different than when we were looking at inheritance within families. In order to identify genetic variants that contribute to risk in an oligogenic or polygenic disease paradigm, we need to compare a lot of patients, in this case Brugada syndrome patients, to a lot of individuals from the general population, what we call a case control design. So what we do, we compare the frequency of thousands of genetic variants spread across the genome between the cases and the controls. So we test thousands of genetic variants, and then we test them all to see whether a specific variant is more present in the cases than the controls, because that would indicate that it is associated with the disease. This we did by means of a genotyping array. So these are products actually that you can buy off the shelf and interrogate these genetic variants throughout the genome. And this is another area where technological developments have helped scientists immensely. And for instance, the most recent array that has been available now costs 25 euros as opposed to hundreds of euros just a few years ago. As I said, the power to detect genetic variants relies now on numbers, especially when you're looking for those genetic variants that have a small effect size. In order to detect them, you really need a lot of patients. No one center in the world can do this alone, so we teamed up with researchers at different centers worldwide, put our patients together to try to tackle this issue. So, to the same, we performed what is called a genome-wide association study, comparing genetic variants in the cases to the controls. And the first time we did this was comparing 300 patients to about 1,000 controls. And this is what you get. This is the readout. This is called a Manhattan plot, or as you hope it is a Manhattan plot, because actually you want to have towers of genetic variants over here, because that would signify an association with a very robust statistical support. So every point, so the numbers over here represent the chromosomes. Each chromosome is represented in a different color, and within each chromosome, each genetic variant that we tested is represented by a dot. And what you see over here is the negative log of the p-value. Now, we're testing over here more than 1 million SNPs, which means that we're doing a lot of tests, which means that we need to correct for multiple testing. And multiple testing genome-wide in such an experiment means that we need to divide the p-value of 0.05 with 1 million, which gives you a p-value threshold of 5 × 10⁻⁸. And that is that red line over here. So what we hope is to find hits that are exceeding that threshold. And as you can see, we identified 2 of them over there. This is more detailed data. So 2 of the hits clustered close to each other on chromosome 3. Another hit occurred on a different chromosome. So this is the data of the discovery set, the initial 300 patients and 1,000 controls. What we have here is the frequency of the risk allele in the cases as opposed to the controls for this marker, for instance. Now, in order to really come up with a robust association, we didn't stop at the discovery set, but found an independent replication set of cases and controls. So now we didn't do the experiment genome-wide, we only tested these 3 genetic variants. We tested these 3 in a replication set, 1 from Europe and 1 from Japan, again achieving very, very robust p-values, and ultimately we meta-analyzed all these cases and control sets together, demonstrating robust association. We also went one step further. So what I showed you before is that we think you need to accumulate these genetic variants to reach the threshold. So we also started looking to see what happens to the risk if we start looking at them in aggregate. So what you see here is the odds ratio associated with the number of risk alleles that are carried. So at every site you carry one variant that you inherited from your mother and another variant that you inherited from your father. So the maximum you can have are 6 risk alleles if you're unlucky, that all alleles that you inherited at these sites are deleterious, or if you're lucky, you inherited only protective alleles. And what you see here, individuals that carry 5 or more risk alleles have an odds ratio of 20 or more than 20 as compared to individuals that carry 1 or less risk allele. Now, this, of course, you need to look with the perspective that this is relative. When one thinks of the incidence of Brugada syndrome, which is only 1 in 2,000, actually the absolute risk that is conferred by these variants is still very small. And that you can also observe from this distribution over here. So what we see here is the distribution of risk alleles in controls, which is in black, and the Brugada syndrome cases. You also see that a large proportion of the germ population also carries 3 or higher risk alleles. So while they have a high relative contribution to risk, because the disease is rare, the absolute contribution to risk is still small. So while we hope that by doing these kind of studies, we try to explain the genetic component which is determining risk in this disorder, again, finding the areas of the genome which are associated with disease, again, gives us some insight into the mechanism of the disease. Now, we were not very surprised to see that actually the chromosome 3 signal is located in the region of the SCN5A gene, where we already knew that rare genetic variants existed. So basically, with respect to the chromosome 3 region, what this experiment told us, that in SCN5A, you don't only have the red variants in the top left quadrant of the graph that I had showed you, that contribute to risk, but in this region there are also common variants with a lower effect that also contribute to risk. However, this experiment also gave us a new region in the genome that we previously had not known was associated with the disease, and that is this one on chromosome 6. And if we zoom on that region, one gene that struck us is this gene over here. It is the HEY2 gene. It struck us because it is highly expressed in the heart. The protein that is encoded by that gene is present in the heart. It also plays a crucial role in cardiac development, in development of the embryo, in the embryology of the heart. But also, we started asking then the question, is it possible that this gene is not only important in the development of the heart, but maybe is also responsible for the homeostasis of cardiac electrical function in the adult? For reasons of time, I won't go into that, but we've subsequently conducted studies on mice that miss this gene, and actually we prove that this gene fine-tunes the level of expression of the sodium channel. So as I said, the variants we've identified contribute very little to risk. There are still many variants out there that we still need to find if we ever are gonna really explain the risk, the susceptibility to the disease. So how do we go about doing that? Of course, one very logical thing is to genotype more patients. So I told you that till now we have done 300 and 1,000. This is what is ongoing. Essentially, we're genotyping everybody that we can find. This is ongoing, but I have some preliminary data but no time for that, unfortunately. But now the real challenge for us is to move from the common variants that contribute a little bit of risk to the rare variants, or let's say low frequency variants that contribute a larger, moderate effect of risk. And here we will use whole genome sequencing, and actually this is the topic of my VG project. And this not only allows us to now look genome-wide for these moderate effect variants, but also allows us to look at the next frontier, I think, of human genetics, which is understanding the role of genetic variation in the non-coding part of the genome. Because till now, we've only been looking at this 1%, and I think really still scratching the surface. So I will leave you with this challenge for the future. So I hope that I've convinced you that we've learned a lot. It has helped a lot, but there are challenges, and this is what is gonna keep us busy in the coming years. And I would also like to take the opportunity to thank our collaborators, which of course are many, because for sure you don't do these studies on your own. Thank you very much for your kind attention.